The next group of lipid-lowering drugs we're going to talk about are the fibrates. Examples of this group include fenofibrate and gemfibrozil. Clue is in the generic names of those two drugs with the fibra in there. These act by activating receptors known as PPARs. So what are PPARs, I hear you ask? Well, that stands for peroxisome proliferator activated receptors, and there are a few of them, and they are denoted by the Greek letters alpha, beta, and gamma, and there's a delta as well, actually. The fibrates are focused on the PPAR alpha. Now, these receptors are very interesting receptors. First of all, they're based in the nucleus, so they're nuclear receptors rather than membrane-based receptors. Now, when you find nuclear receptors, you know that activation of those receptors is leading to changes in gene expression because, of course, it's in the nucleus that the genetic material is held. And so just like the steroid receptors, which are nuclear receptors, these receptors, when they're activated, will cause changes in gene expression and therefore changes in the expression of various different proteins. These receptors as a whole seem to act as a kind of nutritional sensors which can detect whether you're in starvation or feeding mode, and they can alter the metabolism in fairly subtle ways as a result of what your current nutritional status is. So the PPAR alpha that we're talking about here, that is activated by the fibrates, actually allows the adjustment of the rate of fatty acid breakdown to occur, and it increases fatty acid breakdown rates. And we're not going to go in any great detail to exactly how and why they do this, but the next diagram will give you a roundup of how activation of PPAR alpha leads to lowering of lipids. So the fibrates, through their activation of these PPARs, lead to the following changes. Now, let's go through it in a nice methodical way. Let's look first here. You notice that this notation on the diagram says that fibrates increase the expression of proteins in yellow. So all of these proteins that are listed in yellow are upregulated. There's increased levels of those proteins as a result of the changed gene expression that is brought about by activating that receptor. So starting here, we get an increase in this enzyme here, acyl coenzyme A synthase, and you don't need to learn the name of this enzyme, but when it's upregulated, what it means is that you get more breakdown of free fatty acids to yield acetyl coenzyme A. In other words, what we're seeing here is basically more oxidation of free fatty acids to yield energy. So these drugs will increase the burning of free fatty acids in cells, essentially. What will that mean? That will mean that there are fewer triglycerides, because you remember triglycerides will be broken down to yield the free fatty acids. If you're burning up the free fatty acids, then you're going to be using the triglyceride, so you're going to drop the triglyceride level in cells. That in turn will mean that there's less of the raw materials, if you like, to make VLDL. So in turn there'll be a reduction in the levels of VLDL in the circulation. So that's a good thing, because VLDL can cause problems. But if you're making fewer VLDL particles, then it stands to reason you're going to have fewer LDL particles, because LDL comes from VLDL as a result of release of the triglycerides from inside the VLDL. But in addition, you get upregulation of the lipoprotein lipase, that enzyme which actually releases the free fatty acids from the VLDL and leads to the production of IDL and LDL. So you've got less of these low-density lipoproteins, but also they seem to be of an increased size. So there's less LDL, but also the LDL that is there, the particles of LDL are actually slightly bigger. And that means is they're less risky to the blood vessels, because remember, the small size of these particles increases the likelihood of them causing problems in the vessels. In addition, we know that this drug results in increased clearance of VLDL. So not only does the liver make less VLDL, but also a lot more of it gets taken back into the liver and therefore cleared from the circulation. So less LDL, less VLDL, that's all good. And what LDL is left is not as small as it previously was, so that's good. And there's also evidence that the affinity of the LDL proteins for the receptors on the outside of cells is also increased. So again, even the LDL is more likely to be cleared from the circulation. We also see with these drugs an increase in HDL levels. So just like the statins, which are able to increase HDL at the same time as dropping LDL, these fibrates seem to be able to do that too. And they do that by altering expression of the genes that produce these proteins. And these proteins, the apolipoproteins here, are some of those specific proteins that are found on the outside of the HDL particle. So that you make more of those, you can make more HDL and put it into the circulation. And this protein here is involved in the transport of cholesterol into the empty HDL. So if you make more of all these proteins, what you've got is more HDL particles, but also a greater ability for those HDL particles to load up with fats and take them out of the circulation. So from activating this one type of nuclear receptor, you've got increased expression of all of these proteins that are involved in production of lipoproteins and metabolism of free fatty acids. And final result is a drop in LDL levels, a change in the size of LDL particles, and an increase in HDL levels. Brilliant. That's what you need. What are the adverse effects with the fibrates? Well, the common adverse effects, in other words, more than 1 in 100 people might suffer these, tend to be gastrointestinal effects, things like abdominal pains, dyspepsia, that kind of thing. Infrequently, and we define infrequently in this case as between 0.1 and 1%, so less than 1 in 100, but more than 1 in 1,000, will suffer gallstones or pancreatitis on these drugs, and rarely, and that is less than 1 in 1,000, jaundice and anemia. And again, like the statins, but very rarely, you can get myopathies and rhabdomyolysis. So you're not free of that muscle issue even with the fibrates, although again, just like the statins, severe problems are rare. So those are the major adverse effects to watch out for with the fibrates. Now, what about ezetimibe? This is an agent on its own. It's not a class of drugs. So this would be used as a second line treatment for high cholesterol. It would be used for people who couldn't take the statins, had suffered adverse effects from the statins. It acts by inhibiting the uptake of cholesterol from the gut. So it doesn't affect the synthesis of cholesterol in the liver. It acts on reducing the absorption of your dietary cholesterol. So you can see probably straight away that it has less capacity to drop the amount of cholesterol in the blood than drugs that act at the synthesis of cholesterol in the liver. So let's have a look at what happens with ezetimibe. Nice big clear cross showing that ezetimibe inhibits the transport of cholesterol out of the gut and into the bloodstream, which means if we follow that process round that we've looked at before, you will therefore get fewer chylomicrons and therefore fewer chylomicron remnants being taken up into the liver, and therefore less raw materials to make your VLDL, and therefore less LDL as a result of that. So you're simply not getting the chylomicrons made at the gut in the same number, and therefore the liver is not receiving the same number of chylomicron remnants. So LDL is dropped. But there's something else that this drug appears to do which is also very helpful, and that's that same thing that we saw the statins doing, and that is because of the reduced delivery of the chylomicrons into the liver, it stimulates the addition of more LDL receptors on the hepatic cells, which means that it increases the capacity of the liver to pull LDL out of the circulation and into the liver. So it upregulates those LDL receptors, which means that not only is there less LDL being produced in the first place, but there's more being pulled out of circulation. Now, this ability to clear the circulation of LDL at a much higher rate seems to be a really good way of getting LDL levels down. And the new drugs that are in testing and will probably be coming out are going to be targeted around this area, because this seems to be a promising effect. If they can upregulate the LDL receptors on cells, they seem to be very effective at dropping LDL. Now, the last one we're going to talk about is evolocumab. The MAB tells you it's a monoclonal antibody. It's a human monoclonal antibody. We've seen these types of things before, the biological agents. This is an antibody directed against proprotein convertase subtilisin/kexin type 9. Most people refer to that as PCSK9. Now you can see from the A's here that this is an enzyme. PCSK9, if we look over here, is involved when LDL particles lock on to their receptors on cells and then are taken in to the cell in these vesicles. Now, when PCSK9 joins in the ride, which it does seem to do, what happens is that once that vesicle is created and then it fuses with the lysosome, the entire thing is destroyed, which means that includes the LDL receptor. So when PCSK9 is involved in this cycling of the LDL particles, what happens is that you don't get the LDL receptor returned to the cell membrane to pick up more LDL. It just gets broken down completely. When PCSK9 is not present, you get recycling of LDL receptors back into the membrane, which means that you never lose that ability to suck up more LDL and take it out of the circulation. So it stands to reason then that if you can somehow nobble PCSK9, you can make more of your LDL receptors recycled and fewer of them broken down. And that is what that monoclonal antibody is doing. It's targeting PCSK9, knocking it out so that it's not available to get itself involved in this bundling up of the LDL particle and the receptor and breaking the whole lot down. It's effective because, again, it has that very useful action of increasing the numbers of LDL receptors. I'm going to draw some more on the membrane here, and therefore it increases the ability of all the cells affected to pull LDL out of the circulation. The problems with it, it's very expensive and it's a monoclonal antibody, so it has to be injected. It's not able to be given orally. Okay, adverse effects for these monoclonal antibodies. The most common one, which is not unique to this antibody but is shared by all monoclonal antibodies, is problems at the injection site. Reactions, pain, reddening, swelling at the injection site can be a problem, and quite commonly. In addition, you can get respiratory tract infections and flu-like illnesses with this particular monoclonal antibody, evolocumab. Again, they're reported quite commonly, so this is 1% or more incidence. Infrequently, less than 1%, you can get pretty much anything reported. So infrequently with evolocumab, there's been reports of headache, nausea, fatigue, back pain, joint pain generally, muscle pain, infections in other organ systems, even neurological symptoms, confusion, delirium. Pretty much any adverse effect you can think of has been reported infrequently. So this would be something that would only be used if you couldn't use the first line treatment for high cholesterol, which would be the statins. So all of these other drugs, this agent, the fibrates, and gemfibrozil, are drugs that you will see being used as second, third, and fourth line agents for lowering LDL levels in people who, for one reason or another, couldn't take the statins. The statins are still, and will remain probably for some time, the first line treatment to reduce LDL levels. And finally, this is just one of those excellent pull everything together kind of diagrams, which is showing you all of the drugs which can reduce lipid levels in one way or another, including some groups of drugs that we here haven't actually covered, but I haven't taken them off this diagram. I've left them on there just so that you can see that there are a couple of other things, but I'm not going to ask you about them, but I think it's good to have them for completeness. So you'll notice there's niacin here, which reduces lipolysis of fats from adipose tissue, and we've got the bile acid resins, which reduce the reabsorption of bile acids and therefore reduce cholesterol that way. However, you can focus on the drug groups that we've covered, but it's nice to see them all summarized on this one diagram, and I've put this diagram up for you so that you can get hold of it yourself.